I know Matthew asked for us to try and be somewhat punctual because he has a lot he wants to say and wants to keep it in the time. So I'm going to get started. And of course, I want to begin by saying what an incredible pleasure it has been having Matthew in the lab these past years. He's brought so much to both the intellectual breadth of the lab, as well as the culture and and just making us do things. So one thing I want to say about Matthew is he is one of the one of the best instigators that I've ever been around. And I want to say instigator in truly the best possible sense. Whenever we start talking about really good ideas and things we want to do, he always jumps to, okay, well, how do we make this happen? He's responsible for getting us to actually start weekly C++ meetings that a bunch of us go to every week that have been wonderful, even convincing me to turn it into a proper seminar class where he made sure everything worked for the class, which was amazing. During the beginning of the pandemic, when suddenly there was a lot of extra money for undergraduate researchers because so many undergraduate research programs were being canceled when they couldn't be in person. Matthew helped organize the Waves workshop, which brought tons of people virtually to MSU to work on many of our projects. And as we train them in better software development. And there have been so many things that have only happened because Matthew made sure they happened, right? He instigated them and made sure that they went through. And to no one's surprise, he brings a similar motivation and excitement to his research. So Matthew really wanted to study real major evolutionary transitions, and there was so in the natural world, obviously they take a long time, but even to study them computationally, they require both a lot of time, a lot of specifications, and all of the studies that had been done with them, the transitions were forced, and then we studied how they happened. And what Matthew really wanted to do is make these transitions real, and the sheer amount of technological innovation he had to do to make that possible, and the theory that he had to put together in order to figure out even what to build in the first place was really incredible. And so we've had many years here of just intellectual conversations alongside of frustrations on how to get things to work right, and it's all been wonderful, all been a lot of fun. And I'm excited to see his presentation today to really talk about all of the details behind this. So without further ado on my part, I will turn things over to Matthew Moreno to talk about engineering scalable digital models to study major transitions in evolution. So Matthew, over to you.
Great. Thank you, Dr. Ofria, for the kind introduction, and thank you, everybody, for sharing your time with me today. I really appreciate it, and I'm really glad to see you all. I know we have a really broad variety of backgrounds in the presentation today, so I just want to kind of clarify for everybody what to expect up front. I'm really hoping to give a general audience presentation here, so hopefully there's something interesting for everybody, no matter what your background. And there's a special session for me to talk shop with the committee next, so I'm not going to get too technical into the weeds here. And one thing that I've seen work really well in Zoom presentations and Zoom defenses is making good use of the chat to ask like quick clarifying questions. I know there's lots of people here who I've already talked their ears off about all of my research, and so I'm sure they'd be happy to jump in and kind of do quick clarifications as we're moving along. The other thing that I wanted to disclaim up front is that I am using some AI generated visuals as a kind of like fun clip art to keep things interesting. This isn't a topic of my research, but I just wanted to like make it clear up front that some of this stuff in the presentation is generated, and I've labeled everything that's generated as F for fake, so you can kind of, it's really clear what's real and what's not. So just like my last 5 years in graduate school, this presentation is a little bit of a winding journey. And so I want to start off by giving a preview of what you can expect to hear about today. So, and there may be some terms here that you're not immediately familiar with, but during the actual presentation, we'll define them as we go along. So we're going to start by talking about the idea of open-ended evolution and its relationship to major transitions in evolution, which will help us understand why modeling multicellularity is really important in building open-ended evolution algorithms. We're going to talk about how we can use self-replicating programs as a type of evolutionary model system, kind of resembling maybe like a unicellular model. And then we're going to talk about really get into my research question, which is how can we take these self-replicating programs and build digital multicellularity models out of them to study open-ended evolution. And so we'll survey approaches to building models of multicellularity out of self-replicating programs and introduce the dystony model that I've been working with in my own research. Then we will kind of validate the dystony model and look at a bunch of stuff that evolved and see if we're seeing traits characteristic of multicellularity evolving in this model. And finally, we'll introduce some ways to measure complexity, novelty, and adaptation inside of this model system and use a case study along a particular lineage from within the dystony model to kind of take these metrics for a test drive. And then hopefully at the end of this, I might get to graduate. So stay tuned for that. So let's go ahead and jump into the first section here talking about open-ended evolution. So I think we're all familiar with the idea of taking inspiration from nature. Those of us who made it through 11th grade English are all acquainted with Mr. Thoreau and all of the inspiration he got staying at his little cabin by the lake. But nature is also an important source of engineering inspiration in addition to artistic inspiration. I think the invention of Velcro is a great example here, where the hook-like structures on Velcro were inspired by the hook-like structures on the burdock plant that assist with seed dispersal. We can also take more kind of abstract inspiration from nature, and there's a lot of great examples of the kinds of inspiration we can get specifically from the wonderful world of ants, where ants have all these clever ways of making sophisticated decisions as a group without any top-down control. So in this example, kind of figuring out the best place to build a bridge out of their bodies. And by studying ants, we can learn a lot about algorithms, techniques to come up with distributed decision-making results. And so across these examples of inspiration from nature and all the other wonderful ways we can take inspiration from nature, there's one thing in common, right? And that, of course, is evolution, which is the process that gave rise to the functionality and structures that we see in the contemporary biosphere. And we as people have a really rich history of taking inspiration directly from the evolutionary process by replicating parts of it. A good example of this is selective breeding, where we take a wild population and through artificial selection over time, we're able to enhance some kind of trait of interest, whether that's the number of kernels on a cob of corn or trying to make weird fancy pigeons or really buff muscle cows. And there's also a rich history of taking inspiration from evolution within computing. There's a class of problem-solving techniques known as evolutionary algorithms, which you can kind of think of like selective breeding, but instead of making buff cows, we're trying to solve engineering problems. So in this example that I have on the slide, it's from work done by NASA engineers who used a selective breeding approach to try to generate antenna configurations for a spacecraft. And so they started off with a population of antennas, and then through artificial selection over time, they were able to generate these antenna designs that had the reception characteristics they were looking for. This evolutionary algorithm approach, kind of like selective breeding, is a really powerful tool. However, it has some fundamental limitations. One of them being the fact that selection in these evolutionary algorithms is usually directly aimed at solely on the basis of the particular trait that we wish to maximize. And there's been some really amazing work in the last decade from Joel Stanley and Lehman, who have shown that in some cases, directly selecting for a particular trait that you wish to maximize actually isn't the best way to go about things, that you can actually get better results by instead selecting for novelty and diversity, and then kind of eventually seeing properties that you're interested in arise in the system. So the kind of intuition here is that directly selecting for the traits of interest can often get you stuck in evolutionary dead ends where you just keep trying to do the same thing and you're not getting anywhere. Whereas moving away from objective-based selection can kind of help you more effectively explore the evolutionary space and discover stepping stones along the way that eventually lead to the properties you're interested in. And I think the example we were talking about with the burdock plant is a great example to kind of understand the thinking here, where if we step back a little bit, right, this burdock plant wasn't generated through artificial selection. It came out of a natural evolutionary process, and it's not the only piece of the equation. Rather, the structure arose through rich biotic interactions within the ecosystem, right? The hook is only half of the Velcro. There's also all these fuzzy woodland creatures running around that are crucial for the context in which these structures evolved. And so this is where we start to get to the idea of open-ended evolution algorithms, where instead of directly selecting for an objective like we do in selective breeding, we instead try to create a system where we can observe continuing generation of complexity, novelty, and adaptation. Complexity meaning kind of the intricacy and sophistication of the organisms that are evolving, novelty referring to their originality and uniqueness, and adaptation referring to their functionality and fitness. And the idea here is kind of like how we take engineering inspiration from aspects of nature, where we'll just run this system and it'll spit out continuing stream of interesting things for us, and then we can sample among them for inspiration and potential insights. And so this might be a little bit of a hot take, but we're actually reasonably good at the selective breeding like evolutionary algorithms. There's still room for improvement for sure, but in many cases, we're actually able to selectively breed engineering solutions that are competitive with human designs. However, open-ended evolution is a really different story, where within the field, there's a kind of consensus that we're encountering profound difficulties in realizing open-ended dynamics in our simulations. And what's really exciting for me and a lot of researchers who have gotten into this area is that the kind of fundamental insufficiency of our current open-ended evolution algorithms suggests the possibility of major blind spots in our theoretical understanding of the evolution of complexity, novelty, and adaptation in the biosphere around us. And by studying these processes, by trying to kind of recreate aspects of them, that can actually be a really powerful tool to try to better understand natural evolution in a way that isn't always possible by directly observing existing biological systems. And so there's kind of these two avenues of interest in open-ended evolution, right? The idea that we can continually generate novelty, complexity, and adaptation that we can take inspiration from and make good use of, like we do from cool stuff in nature, but also using it as a model system to try to understand evolution. And there's a lot of complementary work exploring different ways to advance open-ended evolution algorithms. However, what I'm going to be talking to you about today are major transitions in evolution. So let's go ahead and define that. So I've got a video of a beehive here where the individual insects in this hive are coordinating to produce these really mesmerizing refractory patterns that it's thought that the shimmering pattern helps to deter predation by causing confusion. But right, so these individual insects are cooperating to perform the sophisticated behavior, but with insect colonies, there's actually kind of an even deeper level of team play going on, where individual insects are happy to, give up their own reproductive interests. Many of these insects are infertile and not generating offspring, and ultimately will often sacrifice themselves for the benefit of the collective. So there's this really sophisticated kind of deep level of team play that's evolving in these systems. And this is really the key idea of major transitions, which is the ability of natural evolution to continually generate new kind of superorganisms, which fundamentally changes the substrate of evolution, moving selection from kind of the level of individual insects to this superorganism collective of this eusocial insect colony. And so eusocial insect colonies are, of course, a classic example of a major transition evolution that fundamentally reshapes the substrate of evolution. But the evolution of multicellularity is also an important example, where we have individual cells coming together in order to reproduce as a group and engaging in really sophisticated behaviors among themselves to make that all possible. And the relationship between major transitions and open-ended evolution is actually pretty intuitive. The idea that the ability for evolution to fundamentally reshape its own substrate over time can be an important way of keeping things fresh and creating continuing opportunities for novelty, right? And this is where we start to get to my own research, which is trying to connect how can we build open-ended evolution algorithms that have the potential for major transitions in evolution. So before we really get into my research, though, we're going to take a step back and do a quick primer on digital evolution techniques and talk about how we can use self-replicating computer programs as an evolutionary model system. So we have to start off by understanding how evolution works. And of course, it turns out that Jeff Goldblum is actually the perfect person to help us understand this. So perhaps no one on the face of the earth better embodies the idea of variation than Jeff Goldblum and all of his wonderful and wacky outfits. And this was a small portion of what I found on the internet before it started to lag at Google Slides. But it's not enough for variation to exist. That variation needs to be heritable. So for the sake of argument, for example, we'll assume that Jeff Goldblum reproduces clonally, and further that the outfit characteristics of Jeff Goldblum are heritable through this clonal reproduction, so that if we have a fancy, formally dressed Jeff Goldblum, generally his clonal offspring will tend to have similar outfit characteristics and not kind of be more ragamuffin dad style Jeff Goldblums. So the other major ingredient in evolution is selection. And there's a very obvious selective effect or selective agent acting on our population of Jeff Goldblums, which is, of course, predation by giant dinosaurs. And it's well understood that razzle-dazzle fancy outfits are an effective defense mechanism against dinosaur predation by causing confusion and panic even among would-be predators. And so we would expect the bulk of the predation pressure to be placed on our kind of more plainly dressed Jeff Goldblums. And so we bring these two forces together, we can see an evolutionary process where we start off with a population of Jeff Goldblums with lots of variability in the outfit styles, and as selection acts on this population, we'll see the composition of the population change. And then because the outfit style is a heritable characteristic, we'll generally see the population shift over time to more and more wacky razzle-dazzle outfits, which is exactly what we see with Jeff Goldblum in nature, of course. So stepping back to how we can use self-replicating programs as a model of evolution, it turns out that self-replicating programs have fundamentally have these two characteristics of heritable variation and amenability to be selected on. And so we can think of these self-replicating computer programs kind of like a computer virus, where it's just a sequence of instructions that when you run them one after the other, it makes a copy of itself and creates another instance of the program in memory. And so these programs can exhibit meaningful heritable variation. So I've got the original program, a simple version on the left, and then I've created kind of a different version of that program with a bunch of gobbledygook in the middle that doesn't really do anything. And these programs will exhibit heritable variation in their efficiency as self-replicators, because this program on the right inherently has to copy a lot more stuff that's not really doing anything for it. It will be outcompeted by the more efficient self-replicators if they're competing for limited space within a computer, because the more efficient self-replicators will tend to better propagate themselves within the population and generally take over. So we have these selective effects and we have heritable variation, and that's what we need for an evolutionary system. All right, this is where I would usually pause for some quick questions, but I think in order to cram everything in here, I'm going to fast forward through this, but I would really encourage people to make use of the chat. And if people want to help out by facilitating discussion, that would be great. So we've talked about how we can use self-replicating computer programs as an evolutionary model. Next, in how those programs, like they in some ways represent single cells. And so the next portion of the talk is going to talk about how we can take this kind of single cell-like model and figure out how to build multicellular model systems out of the self-replicating programs. So the foundational work on this topic has been done by Heather Goldsby and her collaborators in kind of this Deem-based model, where they divide the evolutionary space up into all these little pockets. I've got one pocket as this blue circle on the left here, and within these pockets, there's a certain number of slots that are available for self-replicating computer programs to occupy. And to get things started, we seed a single copy of a self-replicating program into this little pocket of space, and it's going to go ahead and make copies of itself. And once this self-replicating computer program has sufficiently filled up this little Deem area, the Deem itself becomes eligible to create a propagule, and the Deem itself becomes eligible to reproduce. And so we have a population with a certain number of slots available, and once this Deem is full, it can, one of the self-replicating computer programs is allowed to make a copy of itself and found a new Deem, where this process then repeats. So just to make clear what's going on in our analogy here, the self-replicating program itself has information content kind of explaining or specifying which behaviors should occur. So this is kind of like a genome. The individual little virtual CPUs that these programs can occupy within this Deem space are kind of like individual cells. The Deem itself is operating as a multi-cell, and then the population of Deem slots is, I guess, a population. And so there's interesting selective effects going on here where, of course, there's competition within the Deems, right? Where these programs are in some ways competing for space within these little Deem pockets. However, there's also a selective pressure for cooperation within the Deem, because the Deems that cooperate and aren't fighting each other and are able to fill up their little pocket of space the fastest are going to be able to generate Deem offspring the fastest, and then kind of dominate this available population slots for the Deems. And so the system kind of has this fundamental selection pressure for cooperation within these within the Deems, and that's where we see multicellularity evolve. However, there's a fundamental limitation of this. This approach is really powerful and has been used for really great research looking at, for example, the evolution of division of labor and particularly the evolution of soma and germlines. But there's some fundamental limitations here with respect to open-ended evolution, namely that there's no capability within this model for biotic interactions between the multicells, that these multicells are all put in their own little separate buckets and aren't able to have rich biotic interactions like we would see in nature. And so this is kind of where the model that I've been working on comes in. I'm trying to think about, okay, so how can we build on this approach and allow these multicells to interact directly? And so we put all of our multicells in a single two-dimensional kind of flat space You can think of it kind of like a petri dish. And here I've got a single cell with a single genome in it. And we endow these cells with a hereditary group ID, which allows them to kind of build multicellular groups. So when cells replicate in the system, they can create an offspring that has a different hereditary group ID, which is indicated by color here, or they can choose to grow their own group and by placing offspring that inherit that hereditary group ID. And so right, there are these two modes of reproduction where you can grow your own group or you can expel offspring to found a new group. And as we run this forward over time, we'll see these groups grow up and then they'll end up competing for space in the population, which is the fundamental kind of selective mechanism here is the competition for limited space within these petri dishes. And I just want to clarify that, right, the color coding here is this idea of this heritable hereditary group ID. And later on in the presentation, I might just refer to these as groups. And so that's what I'm talking about if I get a little bit more abbreviated. So here's a video of how this works in action. In this video, we have a small number of propagule cells that were seeded into an empty environment. And as time goes on, we can see these cells reproducing and growing up their hereditary groups, and then eventually creating propagules that found new groups, and then ultimately filling up the space and starting to compete for the limited space available within this virtual petri dish. So to demystify a little bit how this is working under the hood, these individual cells are controlled by the exact type of self-replicating programs that we talked about earlier. And these programs are capable of sensing information about their own cell and about their neighboring cells, so about their local environment. And they're capable of making decisions about kind of how and when to reproduce, as well as decisions about, being able to share resources that potentially benefit their neighbors. And even further down under the hood, this is all built using an event-driven genetic programming representation, which the foundational work on that was done by one of the previous grad students out of our lab, and has been really influential in enabling the type of work that I'm interested in. So big thanks to Alex for making this all possible. So we call this model DISHINI, which stands for Distributed Hierarchical Transitions in Individuality. So we've explained how we can build a model out of self-replicating cells. Now we're going to talk about validating the model, right? We have this new model. Now what we want to do is go around and kick the tires, take it out for a spin, and see what it does. And particularly, we want to see whether meaningful multicellular traits evolve, right? Cells that are stuck together are in our case, forming hereditary groups. That is some aspect of multicellularity, but there's a deeper aspect that we're interested in selecting for those fundamental team play mechanics that indicate kind of a fundamental shift in the level of selection. And so what we're trying to do in this section is validate whether we're seeing those team play mechanics evolve. So we generated a bunch of independent evolutionary replicates with some slightly different conditions, and then surveyed them to see what was evolving at the end of evolutionary runs. And across all of our replicates, we saw a significant trend towards reproductive division of labor, where cells at the interior of these hereditary groups were reproducing at a much lower rate compared to cells at the exterior. Which is indicative of kind of the cells at the interior seeding their own immediate evolutionary interests, and the benefit of kind of helping the group itself compete more effectively for space by focusing reproduction on the exterior of these groups. We also saw other characteristic traits of multicellularity occurring within the groups. We saw resource sharing behaviors emerge across a lot of our replicates, where cells were specifically sharing resource with other members of their group in order to facilitate the fitness of the group. We saw, interestingly, resource investment in offspring groups. So when we have one of our hereditary groups and you create a propagule offspring that founds a new group, we can refer to that as an offspring group. And this is kind of a basic form of parental care where the parent group is sending resource to its offspring group to help it grow up to a size where it can be self-sufficient. We saw asymmetrical behaviors mediated by cell-cell messaging in across some of our replicates. And so this was mostly revolving around resource sharing, where cells were forming these non-trivial resource sharing networks within their groups by basically talking to each other and incorporating that information into to building the resource sharing pattern. We also saw some morphological patterning in some of our replicates. And interestingly, in 2 of our replicates, we saw programmed cell death evolve, where cells were intentionally destroying themselves to benefit the collective. And we were able to confirm through competition experiments, through fitness experiments, that this programmed cell death was actually beneficial for fitness in these 2 cases. So having seen lots of traits characteristic of multicellularity evolve in the system, we were interested in kind of getting a more deeper intuitive understanding of exactly how the life histories of these groups are unfolding in the simulation. And so we went through and qualitatively categorized some of the different life histories that we were seeing. First one I'll talk to you about today is this kind of sweep life history. So in this little image up in the top left, I have kin groups color-coded in grayscale, and we have 2 that we're focusing on. The one in purple is a parent group, and the one in orange is the offspring group. So it was created as a propagule from this parent group, and it's starting off, as a single cell. And as we run evolutionary time forward, we can see this offspring group grow back into the parent group, and slowly but surely grow back and take over the space that had been occupied by the parent group. And so here's a video animation of kind of how this looks. I've highlighted the peripheries between parent groups and offspring groups in red and yellow here. And so as the simulation runs, we can see offspring being generated on the periphery of groups and then growing back over the parent group to take over the space that had been occupied by the parent. Another life history that we saw evolve was a burst life history, where instead of offspring being founded on the exterior of groups, offspring groups were being generated at the interior of the group. So here's a parent group in purple, a generation of a offspring group in orange, and then over time, this offspring group, along with several other offspring, grow up and ultimately destroy the parent from the inside and kind of take over that space from the parent. So here's a little video animation of how this looks in the simulation, where you can see these individual little propagules being founded at the interior of groups and then growing outwards to destroy the parent and kind of take over that space. Finally, not all of the life histories that we saw in the system were as destructive of a relationship between parents and offspring. We also saw life histories evolve where offspring were created at the exterior of these groups and then kind of grew outwards from the parent group while leaving the parent group intact. And so here's a little video animation of how this looks in the system in one of our replicates, where you can see these stable interfaces between parent groups and offspring groups and competition for space occurring at the periphery of these kind of stable interaction networks of related groups. All right. So the last thing that I have for you today is looking at a case study of a lineage from within this Dishony simulation system. And the like strategic goal here was to develop and test out ways to measure those fundamental building blocks of open-ended evolution, complexity, novelty, and adaptation in the system. But there's also some interest in kind of understanding how these traits change over time along our evolutionary lineage. So to do this experiment, we performed 40 independent evolutionary replicates where we seeded our Petri dish with randomly generated programs and then ran them forward for little 3-hour windows of evolution. And we used 4 CPUs, 4 threads cooperating to actually perform the computation over this 3-hour window. And then at the end of the 3-hour window, we snapshotted the entire population, saved it off, and then started things up again and ran forward for another 3 hours. And we performed 100 of these little windows of evolution, which we refer to as stints. This was sufficient to observe about 20,000 cell generations. And then for a case study, we went through our 40 independent replicates and looked for something interesting, which we'll be talking about next. One other detail that will become relevant in a few slides is we used a diversity maintenance mechanism to ensure the long-term coexistence of 2 completely independent lineages within our Petri dish. And so one of these lineages we arbitrarily selected as like the focal strain in order to like do all of our studies and do all of our characterization on. However, there was a second strain that was coexisting in that same space with our focal strain and interacting with it over evolutionary time. And the idea here was to promote diversity of biotic interactions within the system by ensuring this long-term coexistence. So first thing we did with our case study was to try to get a qualitative understanding of the life histories that we were seeing evolve in these multi-cells. And so we took sample genomes from each of our 100 time points and grew them up in a monoculture and recorded little videos of their life histories, and then qualitatively categorized them based on the characteristics that we were seeing in those videos. And so we came up with 10 kind of qualitatively different morph categories that we observed over the course of this evolutionary run. And I'll talk to you about some of the highlights. So at the beginning of the evolutionary run, we saw, kind of as you would expect, no formation of these hereditary groups, that cells were running around and competing with each other kind of on a unicellular basis. We refer to this as morph A. At time point 14, we saw the evolution of kind of generally uniformly sized but irregularly shaped hereditary groups. And we refer to this this life history as morph D. And then at time point 15, we saw kind of a first really interesting novelty along our lineage, which was, where's my mouse? Oh, here it is. Which was the evolution of a kind of cigar-like body plan. So here, the hereditary groups are being shown as color-coded, and so these individual streaks of color across the screen are these horizontal body plans that these groups are growing up in the system. And so we refer to this as morph E. And then later at time point 45, we saw a secondary novelty where these cells were still forming these kind of cigar-like body plans, but on an apparent internal timing cue, which you just saw right now, they engaged in a burst of secondary growth along the vertical axis. And so they kind of added a new stage to their life history. And so we refer to this as morph G. So seeing these qualitatively distinct morphologies evolve along our evolutionary lineage, we one of the first questions we had was the extent to which these morphological innovations were adaptive. And so in order to measure fitness and adaptation in our system, we use competition experiments. And how these work is we took a population from the end of one of our little evolutionary windows and from the beginning of the window, and then competed those two groups against each other within a fixed size environment. And if we saw no change in the population composition after running this forward and letting them duke it out, we can conclude that there's not a big difference in fitness between these two populations. However, if one population takes over and excludes the other population, then we can deduce that that population is more fit. So you can think of this experimental technique kind of like Rock'em Sock'em Robots, where we're trying to understand how these the strength of these organisms at competing for space by directly matching them against each other and seeing who comes out on top. So here I have the magnitude of fitness change over each evolutionary time point as a heat map. Time starts in the top left. So this is stint 0, the very beginning of the evolutionary lineage. Time goes to the right across the top and then loops back down. So we go over to stint 50 and then loop back down and keep going along the bottom right, and then end with stint 100 in the bottom right here. The color coding and the lettering refer to these different morphological categorizations that we came up with, which category of morph we were observing at each time point. And I've highlighted here where we first saw those cigar-like body plans evolve, and then where we saw that burst of secondary growth evolve. And there's some, oh, right. And the other thing, of course, is talking about how fitness is shown on this diagram. So this is a heat map. And so blue color indicates a fitness gain over that evolutionary window, with a deeper, darker blue indicating a stronger fitness gain. And red indicates fitness loss, with a deeper, darker red indicating a stronger fitness loss. So some stray observations here. We can see generally kind of more dense, strong fitness gain towards the beginning of our evolutionary experiment, although the single largest fitness change we detected at stint 93 near the end of our experiment. Interestingly, the stints where we saw the emergence of these qualitative morphological novelties didn't have fitness changes that were particularly strong or exceptional with respect to contemporary evolutionary windows where we didn't see novelties evolve, although both of these novelties were associated with adaptation. And then the other thing that we saw was whole population fitness loss appearing to occur along this lineage. And so we did a little more statistics, and here I've color-coded our heat map just as whether there was a significant fitness gain or a significant fitness loss. So loss in red, gain in green, no significant change in yellow. And as we can see here, there were 16 of these evolutionary windows, these stints, where we saw significant fitness loss across the whole population. And this is kind of a surprising result, and not something that there's mechanisms that can lead to this kind of this kind of outcome, but not something that you would generally expect to see in an adaptive evolutionary process. And this got us wondering whether our fitness assay, the technique we were using, was really telling the whole story about fitness. And in particularly, we started wondering whether that background strain was having an effect on fitness. And so what we did was adjust our fitness assay methodology, which had previously just had the foreground strain and added the presence of the background strain into the environment. So we're still competing red versus blue, but now green is also mixed in and hanging out. So you can think of this kind of like adding an audience to our Rock'em Sock'em robot competition and seeing whether the presence of this audience is having an effect on the outcome of the competition. And so we started off with 16 whole population fitness loss events. And when we did the measure without the biotic background, and once we added this biotic background in, we saw only 3 whole population fitness losses. And control experiments suggest that part of this reduction in the observation of whole population fitness loss is related to reduced sensitivity of the assay with the addition of this biotic background. But that does not explain our observation of 6 whole population fitness gain outcomes with the biotic background, where without the biotic background, they had previously looked like fitness losses. And so this is a really strong indication that this background strain is having a rich selective effect on our focal lineage that that some measure of adaptation along this focal lineage is directly related to interactions with that background strain. And so that is really exciting and interesting to see. So having looked at adaptation, next we wanted to look at complexity. We have 2 measures of complexity that we explored. The first measure is sequence complexity. We tried to measure the complexity of the genomes over our evolutionary lineage. And so we're trying to count up the number of sites in our genomes that actually do something useful and contribute to fitness. And so you can kind of think of this process like a little bit like playing with a Jenga tower, where we tried taking out individual blocks, disabling like individual sites in the genome and seeing which sites we could disable without a fitness effect, and which sites, when we disabled them, the Jenga tower came crashing down and we saw fitness loss. And this gives us a way to count up the number of sites that are contributing to fitness, right? By going through and pulling out all the Jenga blocks one by one and seeing how many are actually doing something structurally. So I have some pocket slides I'm happy to talk about in the Q&A with some more details about how exactly, like some of the finer details about how we did this. But without further ado, here's the trajectory of sequence complexity that we measured over this evolutionary lineage. So I have time on the x-axis going from the beginning at 0 up to time point 100. On the y-axis, I have our sequence complexity measure. So how many sites in the genome are actually doing something interesting. And the color coding and letters refer to the different qualitative morphological categorizations that we came up with. And I've highlighted the dotted line is where we first saw the cigar body plan, and the dashed line is where we first saw that burst of secondary growth. So some interesting observations here. Actually, let me go back. So sequence complexity started at about 5 sites at the beginning of the simulation and then grew up and then had a big jump within kind of our the parts of the lineage that was expressing morphology B up to around a little less than 30 sites. The emergence of morphology D was associated with another big increase in genetic complexity. But interestingly, the emergence of morphology E, where we first saw that cigar body plan, wasn't associated with as large of a increase in sequence complexity. And phylogenetic analysis of kind of these populations suggests that morph D and morph E are both directly descended from morph B. And it's not the case that B evolved and then D was descended and then E was descended from D. So D is kind of like a little interesting dead end that eventually went away. For the next about 20 stints of evolution, we saw a more continual increase in sequence complexity. And then the emergence of morph G wasn't associated with a big change in sequence complexity. And then over the remainder of the experiment, we saw kind of a stabilization and tapering off a little bit of sequence complexity, likely as these genomes kind of pared down a little bit to increase maybe mutational robustness. So we are also interested in measuring complexity in terms of the richness of interactions that these genomes were having with their environment. And so we did an additional complexity measure to try to measure that precisely or directly. So there's 3 different ways that genomes in our system can interact with the environment. They can send arbitrary messages to other cells. There's kind of a set number of output dials that they can control to tell the environment what they want to do to control their phenotype. And there's a fixed number of kind of input sensors and input signals that they receive from the environment, telling them what's going on and allowing them to kind of sense their surroundings. And so the question here with these different types of ways that cells can interact with their environment is how many is DJ genome using in order to do something important for fitness? And so I have some pocket slides about like exactly how we measured them. And I'm sad that I don't have the pocket slides because it's a Freaky Friday based metaphor, but suffice it to say that we basically went through and turned off each of these possible interactions one by one and saw whether there was a decrease in fitness. And this is the trajectory of interface complexity that we saw over our evolutionary lineage. Time, again, is on the x-axis. On the y-axis is our interface complexity measure. And I've highlighted here again with the little lines where we first saw that cigar body plan and then where we saw that secondary innovation of that kind of burst of growth on the internal timing queue. And so this is telling a little bit of a different story from sequence complexity, where morph D, which had been associated with a big jump in sequence complexity, wasn't associated with a big jump in interface complexity. Conversely, morph E, which hadn't been associated with a really big jump in genetic complexity, was associated with a nearly tripling of interface complexity. And then morph G wasn't associated with a change in interface complexity. So there's some interesting anecdotal takeaways here. Where we can see, we're seeing novelty, complexity, and adaptation within the system, but they appear to be somewhat loosely coupled, where novelty is not yielding exceptionally strong fitness effects. Novelty is sometimes associated with large changes in complexity, but not always. And we're seeing at times kind of opposing signals from sequence complexity and interface complexity, which really emphasizes the importance of taking a really broad view of complexity and not trying to simplify it and boil it down too much. So now we get to the conclusion part where we wrap things up. There's 4 main things I want to touch on here. I want to give you a recap because I know I've been shooting a lot at you. Talk about some of the key research results, talk about other topics I didn't have time to talk about, and talk about future work. So just to recap what we talked about, we introduced the idea of open-ended evolution and talked about its relationship to major transitions, which helped us understand the importance of modeling multicellularity and open-ended simulations. We talked about how we can use self-replicating computer programs as evolutionary models. We talked about how we can build models of multicellularity out of these self-replicating programs. We validated the dystrophy model, and seeing that we are selecting for meaningful multicellularity. And we did a case study of complexity, novelty, and adaptation along one of our lineages in order to kind of test out these and develop and test out measures for these phenomena. So the key results here, right? My fundamental research question is how can we create digital multicellular models with rich biotic interactions to study open-ended evolution? We were able to confirm that characteristic multicellular traits are evolving dystrophy, notably reproductive division of labor and resource sharing. We were able to confirm that rich biotic interactions were occurring between multicells, where we saw in some experiments parental support for offspring groups, so interaction between those 2 groups, as well as this really interesting sign change fitness effect of a background lineage in our case study. And we were able to confirm that we can use this model system as a tool to study open-ended evolution by demonstrating how we can measure complexity, novelty, and adaptation in the system. There's a bunch of other stuff, like basically half of my dissertation isn't in this presentation today. I'm also really interested in thinking about how we can scale up these types of simulations by using high-performance computing clusters to bring more computers in and simulate larger systems. And also, when we have these evolutionary systems running around our compute clusters, how can we track the evolutionary history in this kind of distributed environment? So I'd really love to talk about these with anybody who's interested. So find me, shoot me an email, whatever. And with respect to future work, one thing I'm particularly excited about is this observation of the sign change fitness effect of the background strain. And I think it would be really interesting in particular to rerun the complexity metrics in the presence of the complexity assays in the presence of this background strain and see if there's aspects of complexity that we weren't able to detect before without this biotic context. And it would be really, really cool to be able to directly attribute parts of this complexity to interactions with that other lineage. And now I think the most important part of this presentation, this research in my own career really wouldn't be possible without a whole lot of help from a whole lot of people. And it really does take a village to make this all possible. I want to really thank my mentors in science before I entered graduate school, who had a really big impact on helping me develop my own research interests in biology and then biological modeling, as well as getting me engaged and really excited about the culture of science and really making me feel at home in this type of environment. During my time in graduate school, I've had the opportunity to work with a bunch of great undergraduate students who have made really direct contributions to the research I've been talking to you about today. I especially want to call out Santiago, who has been with our group for like 4 years and has been a major asset and graciously jumped in the trenches with me in my final push to get my dissertation over the finish line. Of course, a big thank you to my lab group for the stimulating and rewarding environment. A big thank you to my committee and advisor. You know, what hasn't been said about Dr. Ofria and dissertation acknowledgements, but I particularly want to thank you for the unconditional support you've shown me. I remember during one of our first meetings, you made it really clear that if I ever ran into any issues, that, you know, with anything in academia, I should feel free to CC you, and you would jump in and help do battle to take care of things. And I knew I was in a really good place after that conversation. And of course, my committee, I'm not just talking to them now for the first time. They've been giving me guidance along the way and helping me focus and improve my dissertation and, you know, not deforest the world by creating a print copy. And none of this would be possible without the administrative staff who make everything happen and make problems magically go away. Connie James and Melissa Williams have been particularly impactful on me over my graduate career. And of course, a big thank you to my friends, family, and loved one for your support along the way. And I'll go ahead and take questions now. Thank you.
Well, thank you, Matthew. And yeah, we should take time for a few questions from the non-committee, if anyone has any.
Got a question, and I can't figure out how to raise my hand in here, so I'm just gonna
Hi,
go for
Ryan.
it. Hey, can you go back to your fitness assay slide?
Yep. The one with the biotic background or without?
The comparison of with and without. Yeah, so I was wondering, oh yeah, right.
Yep.
Like forward a couple, but yeah, I was wondering if you only looked at the timestamps where you'd seen a negative change in fitness and compared it with the background, or if you also compared the positive and neutral ones, and if you saw any that when you added in the background weren't increased fitness.
Yeah, that's a great question. And we did run this assay across the entire history. And that data is in the dissertation. I don't remember off the top of my head. I believe we did see maybe one case where in the presence of the biotic background, things that had looked either as a gain in fitness or neutral were actually deleterious. but we didn't see as many of those as we saw the opposite way, where adding in the biotic background was causing a increase in fitness. Is that kind of what you're asking about?
Yeah, that's exactly what I was asking.
Yep. And one thing Santiago pointed out to me yesterday that I don't know off the top of my head that would be really interesting is seeing where these, like when and where these biotic events that are, where the background strain is having the sign change fitness effect, how that fits into the, like where that fits into the evolutionary lineage in terms of the other stuff that's going on.
Yeah, it looks like Cliff has his hand up.
Yeah, I have 6,000 or 7,000 questions, but I will limit myself to the 3 that I wrote down, and I'll try to be quick about them. Really phenomenally good work.
Thank you.
Better than expected. Well done. So the first question, relating to the question that was just asked, and sorry to get a little linguy, but do you think that there's potentially Red Queen effect happening between these 2 strains, even within stints? So like you'd have to look at like the actual step-by-step transitions through the whole stint to see if there was some, interaction.
Could you refresh me and everybody else here on like the basics of the Red Queen effects?
Sure. So Red Queen is this idea of like running in place. So if you've got 2 a lot of times it's in sexual selection, but if you had 2 species, one of them makes a change that forces the other one to make a change.
Yep.
And that makes another change.
I see. Yep.
And like at the end, you're worse. Yeah.
Yep. So in terms of the deleterious outcomes that we saw, that's definitely a possible explanation. And that type of interaction is definitely happening in the system. In some of our early experiments, we were looking at like trying to figure out why we weren't seeing apoptosis evolve when we expected to see apoptosis evolve. And what we figured out was happening was under the hood, these programs are organized into little modules that can be activated by signals. And when cells send a message to another cell, they activate a certain little module within that other cell. And the cells that apoptosis wasn't evolving, because as soon as it evolved, all the other cells would figure out how to send messages that would activate that and then basically, cause that cell to self-destruct. And so it's like kind of a stop punching yourself kind of a dynamic there. And so we added in the ability for cells to filter the incoming messages and like decide what they were actually which parts of their genome they were actually going to let be triggered by their neighbors. And in other work, I kind of generated a bunch of different lineages and then did a set of competitions among them and then looked for cycles, like kind of rock, paper, scissors, where A beats B, B beats C, C beats A, and was able to see some of those cycles in terms of, right, so fitness in the system isn't, it's not unidimensional, it's fundamentally like who you're around and that we can see those kinds of effects. Does that get at the question, Cliff?
Yeah, no, and I think what you just said at the end, that your fitness is the product of your environment, which is the other agents. So
yep.
Okay. Yep. So related on this question of communication, you might have said it.
Yep.
Did you allow for the sharing of resources between propagals, and if so, did you see parent to offspring resource sharing?
Yes. So under the hood, these cells in our model, they can't reproduce whenever they want. They have to accrue like enough of a little virtual resource, and then they can spend that to create an offspring. And we allowed cells to share resource with any neighbor cell that they so desired. And we did see some instances where parent groups were just dumping resource into their offspring groups in order to help them ostensibly grow up to size. And when we turned off that particular aspect of resource sharing, we saw a decrease in fitness.
Awesome. Okay, cool. Last question. this growth in the morph E.
Yes.
Like there's a long period where morph E dominates. Do you think that there was the potential, and there's sort of an increase in diversity potentially during that period? Do
Yeah.
you think that that could have been drift because there was little competition because it was all E?
Yes. Yep. So what's interesting is, so we have that background strain, and part of the data that we collected was, so in order to maintain the background strain and the focal strain, we basically penalize whichever strain starts to take over too much of the population. And looking through the data, there's actually, during that period, the back half, a big jump in, instead of having the focal strain have to be penalized because it was crowding out too much of the background strain, it actually flipped a little bit, where the background strain had to be penalized because it was outcompeting the focal strain. And so I mean, that's an interesting aspect of competition that changed during that kind of second half of evolution. So I think I'm not sure about kind of competition within the focal strain, but there were other things also going on that I do have like a little bit of data suggesting that there's something going on.
Cool. Well, thank you.
Okay, and you have a couple of questions in the chat. I think they'll both be quick, and then we should probably switch over to yeah.
Yep. Oh, let me. Could you read them to me?
Oh yeah, I can read them to you if you want. Sorry, I assumed you had the chat open.
Oh, okay. Ryan asked, was the background population also allowed to evolve? Yes, the background population was completely identical to the foreground population, and we basically used a arbitrary mechanism to select which one was going to be the focal one.
And then Austin,
yeah. Sequence complexity, yes, we did measure that. I don't know if I have that in my pocket slides, but
for those who haven't looked at chat, the question is, while sequence complexity was changing, did overall sequence/genome length change?
And I think I had the foresight to put this in here, but nope. No, I apparently did not. So we did see an increase in genome length occurring, where it grew up to about 1,000 sites, and then kind of started growing more gradually. And then interestingly, that huge fitness increase we saw at Cint93 was a big jump in genome length, which indicates that there was probably some kind of like a genome partial duplication event that occurred at that particular time point. And so, yeah, the genome length, I believe, was growing really fast during like the first 20-25 stints, and then it was still growing a little bit and then kind of tapered off. And then at Cint93, we saw a big event where it jumped. That's a great question. Thank you, Austin.
Okay, why don't we all thank Matthew again, and then we will switch over to the private.
Thank you, everybody. I was really glad to see everybody today. Thank you.